Hello, everyone, and welcome to today's webinar, Unveiling Brain Complexity with the MERSCOPE platform using 1000-plex gene panels, I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Vizgen. To learn more, visit vizgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speakers, Peter Reinhold, Global Product Manager, Assays and Reagents at Vizgen. Also joining us today is Dr. Renchao Chen, Application Development Scientist at Vizgen. For a complete biography on our speakers, please click on the Presenter tab. Peter, I'll pass it over to you to get us started.
All right, thanks, Kaylee, for that introduction. So really excited today to be talking about the MERSCOPE 1000-plex gene panel solution that has recently been launched as of last week on March 5th. And so very quickly today, what I'll be talking through is give a quick overview of MERFISH chemistry, an overview of the MERSCOPE platform, and then give some new details on the new product introduction, the MERSCOPE 1000-plex solution. From there, I'll hand things over to Renchao Chen, who's an application scientist who's generated some truly impressive data leveraging the 1000-plex gene panel solution in both human and mouse brain samples. So here at Vizgen, we use MERFISH chemistry to generate spatial transcriptomics data. MERFISH stands for multiplexed error-robust fluorescent in situ hybridization, and uses combinatorial labeling, sequential imaging, and error-robust barcoding to detect, quantify, and localize transcripts from a number of genes across an entire tissue section. MERFISH itself has been around since 2015, coming out of Harvard, and has definitely contributed to generating a lot of buzz around spatially resolved transcriptomics, with Nature Methods naming it Method of the Year in 2020. Now, together with MERFISH chemistry, what Vizgen has designed is the MERSCOPE platform. And this is an end-to-end platform for performing MERFISH in your laboratory. So where this process begins is in our custom gene panel portal. Here, customers can upload gene lists that are important to you for your research into the portal and design a gene panel solution that's going to answer the complex questions you're after. From there, Vizgen will manufacture that panel and ship it to you for use with our comprehensive suite of reagents and assays within the sample prep workflow to prep your samples for downstream MERFISH analysis. We combine those two components on the software and consumables side with our MERSCOPE instrument, which automates the sequential rounds of imaging and the data acquisition that are necessary for generating the MERFISH detection. Once that data is acquired, that's uploaded into our image processing solution, and a number of files are created, including a VZG file, which allows users to visualize the data set that they just analyzed on the MERSCOPE instrument. And so with both the MERFISH chemistry and the MERSCOPE platform, you're able to profile gene expression in situ across large tissue areas. And so here on the left, we're just showing 8 RNA subsets across a mouse brain tissue section. And you can see that we're able to spatially locate those transcripts across that entire tissue section and assign the locations there. The impressive part about this is we can also zoom in subcellularly down to individual cells, look inside those cells, assign those transcripts there. We can do this because we have a cell segmentation algorithm that really complements our entire platform. So this is important as once we assign those transcripts into those cells, We can then identify them and then map those cells back to the entire tissue section and get an idea of where those cells exist on a tissue level scale and which cells they're next to and which cells they're interacting with. In addition to getting that spatial data, what's also important is generating quantitative data and highly reproducible data as well. And so we have benchmarked this against an adjacent technology, so bulk RNA sequencing. So if we look at the graph here on the left, you can see the copy numbers of cells, the copy number per cell that are detected with MERFISH compared with bulk sequencing. You can see we get a very strong correlation between those two. In addition to generating quantitative data, we always want to make sure that that's highly reproducible. As you can see here, two technical replicates. If we look at that correlation, it's very high at 0.99. And so combining that highly accurate, reproducible, and spatial data, it gives users the ability to look not just at the whole tissue section, within specific tissue areas, down to subcellular areas as well. And customers have been using this and demonstrated its use across multiple different tissue sections and species, right? So we've proven that across 30 plus tissue types. Customers going after cell atlas analyses, tissue composition analyses, and even disease progression and pathology. And so up until this point, Vizgen has offered a 140 gene panel, a 300 gene panel, and a 500 gene panel solution. But we have now pushed that limit up to 1,000, and we are extremely excited to announce that we now have the MERSCOPE 1000-plex gene panel solution. This 1000 gene panel is fully customizable. That means you get to choose which genes go into it. You get to choose what's important to your experiment. This new 1000 gene panel will complement our current portfolio of gene panels, so we will continue to support 140, 300, and 500 gene panels. What this means is it just gives you flexibility in choosing the solution that works best for what you are trying to do. The two physical components of the solution include the MERSCOPE 1000 gene panel, pictured here on the left, and the MERSCOPE 1000 gene imaging kit, pictured there on the right. Both of these are now available for order. And the best part is designing and running a MERSCOPE 1000 gene experiment is very similar to our current gene panels, designing and running those on the MERSCOPE instrument. Again, all of this starts with our gene panel portal, which has been updated to allow upload of up to 960 genes onto the portal. We've also, with that, increased the abundance limit for panels to support those larger panels being built. Once that panel is built and finalized, it then gets manufactured by Vizgen. We then ship that panel to your laboratory where you can use that panel in the sample prep procedure. So similar to our current panels, this is the same procedure. You're incubating that gene panel, or that 1000 gene panel on your sample in preparation for running it on the MERSCOPE instrument. Once that sample is ready, we'd move over to the instrument, and together with loading that sample onto the instrument, you would use our MERSCOPE 1000 gene imaging kit in a similar fashion to our current imaging kits. So very simple and easy to use, especially if you're already running MERSCOPE experiments. And we're confident that the MERSCOPE 1000 gene panel is going to allow you to do more. From unraveling complex biological processes, so the added flexibility gives you the opportunity to target more biological pathways. Or if you're after deeper cell subtyping, this is going to, added flexibility here is going to allow you to include additional cell markers so you can further characterize those diverse cell populations. And really this all comes together to just have added flexibility for you as a user to design what you want to do with this solution. This added flexibility gives you the power to answer tough biological questions, all within a single MERFISH experiment. And of course, all of this is built on the MERFISH chemistry and technology backbone. And that's going to allow you to generate comprehensive spatial genomics data sets for your research, and again, answer those tough questions that you're after within your application area. And to demonstrate some of that applications data and what you can do with the 1000-plex gene panel solution, I'm now going to turn things over to Renchao Chen, who's our data application scientist, and he will walk through some of the exciting data he's generated on both human and mouse brain samples.
Thanks, Peter, for introducing our MERSCOPE platform and the new 1000-plex gene panel. Hello, everyone. I'm Renchao Chen, application scientist from Vizgen. Today, I'm going to present two showcases generated with the MERSCOPE 1000-plex platform, one from mouse brain and the other from human brain. Hope can give you some insights on how the new 1000-plex panels can help you understand the brain complexity in both health and disease. So, before diving into the data, I would like to briefly talk about why spatial information is critical for understanding the brain function and dysfunction. So why spatial information is important? Because biological systems are intricately spatially organized. Take the brain as an example. It's not a random mixture of various cell types, but a highly organized structure with all cells organized spatially and connected. With single-cell sequencing, you can obtain molecular features of each single cell or cell types, but that doesn't automatically lead to a good understanding of how the tissues and organs organize and works. On the other hand, many conventional histology work, such as staining, in situ hybridization, tracing, have generated a lot of knowledge on tissue structure, but with limited molecular information. Fortunately, with a cutting-edge spatial genomic method such as MERFISH, now we are able to bridge this gap. By obtaining comprehensive molecular and spatial information at single-cell resolution at the same time, we're not only able to draw bigger and finer pictures of the tissue organization, but can also get functional insight from the data such as cell-cell interaction and tissue microenvironment in health and disease. So here are some examples of how spatial genomic approach alone or combined with other methods can help us to understand the brain structure and function. First of all, they can be used to identify molecularly distinct cell types and their spatial organization. This work published in last year from Xiaowei Zhang's lab generate a whole mouse brain cell atlas with MERFISH, which potentially provide a fundamental and universal framework for molecular and cellular study in mouse brain. Secondly, spatial genomic approach can help map the connection of different cell types. This work developed by SEQ by combining viral tracing, new generation sequencing, and in situ sequencing, which enable high throughput connectivity mapping of molecular distinct neuron types. As another example, this work combines in vivo Cas image and post-hoc multiplex fish image, which is a very powerful approach to link the activity of molecular defined neuron types to certain animal behavior. Last but not least, spatial genomic approach can advance our understanding of brain disease. In this work, we also perform spatial transcriptomic and in situ sequencing assay to understand the cellular and molecular changes associated with the AD hallmark, A-beta plaque. This could not be achieved without spatial information. So here, I'm going to briefly introduce how the MERFISH technique works in case you are not already familiar with it. So MERFISH stands for Multiplex Error Robust Fluorescent In Situ Hybridization. In principle, MERFISH is based on single molecule fish, which use multiplex fluorescent probe to target a single RNA species. After hybridization, single RNA species are represented as dots, and you can quantify the mRNA copy numbers by counting the dots. Although single molecule fish is a quantitative approach with high detection efficiency, its multiplexing capability is quite limited. To achieve high multiplexed RNA detection, the in MERFISH design, each gene to be targeted or each gene to be detected is assigned a unique barcode such as 100110000. The encoding probe without fluorescence signaling are first hybridized to the RNA target, with probe target different RNA species with a handover that could be bound by a secondary readout probe. Then, in different image round, different readout probes with a certain fluorescent signal bind to the encoding probe, and the images are taken by microscope. In a certain image round, 0 means no fluorescent signal, while 1 means a positive signal was detected. After completing all the n rounds of image, we assign the barcode to the decoded probe and identify those genes that sports binary barcode corresponding to. In this way, hundreds to thousands of RNA species could be profiled in the same experiment, making it a leading technology of spatial transcriptome. Okay, let's go to the more interesting part of today's presentation, how the data look like. In the mouse brain showcase, I prepared 17 fresh frozen mouse brain coronal sections with a 1000 gene panel designed for mouse brain, including both cell typing and cell-cell signaling. After MERSCOPE image and data processing, I combine the gene expression matrix from all the samples for downstream analysis, which provides insights into the molecular and cellular architecture of the mouse brain. So after obtaining the MERSCOPE data, first I did some benchmark analysis to assess the quality of the data. When comparing the dataset generated from two mouse slides close to each other, we got high consistent count number across all genes. The Pearson correlation coefficient between the two experiments is 0.99, demonstrating good reproducibility between experiments. By comparing the MERSCOPE data to bulk RNA sequencing data of the mouse brain, we can see a good linear relationship between the two methods, supporting high accuracy of morphology measurement. To assess the detection efficiency, we compare the count number of the 1000 plex gene panel with the 500 plex gene panel, which have around 200 genes overlap. Again, you can see a very good linear relationship, and the ratio of counts for corresponding genes is also very similar, demonstrate a minor decrease in sensitivity when doubling the gene panel size. To further test this, we plot the overall transcriptional density across different gene panel sizes from 140 to 1000. So here, the results show a good linear relationship across different gene panel sizes, also suggest that the sensitivity of 1000 plex gene panel is maintained compared to previous panels. So in addition to assess the accuracy and the sensitivity of the data, we also compare the spatial gene expression pattern from MERSCOPE with those obtained from conventional in situ hybridization. Here are some examples. As you can see, the spatial pattern of the same gene between MERSCOPE data and the in situ hybridization are highly consistent with each other, further confirming the accuracy of the data. So for downstream analysis, I combine all the cells from 17 samples, got almost 1.8 million cells. The first round of cluster analysis revealed 34 cell clusters, which exhibit distinct gene expression patterns, as you can see here. By combining both gene expression and spatial information, it's relatively easy to annotate cell cluster. As an example, here I plot cluster 23, which is a highly expressed marker GPR88. So when plot the cell cluster distribution and the marker gene expression on brain slice, we can see these cells are highly enriched in stratum. With the gene expression and spatial information, we can annotate this cell type as median spanning neuron. So to understand the relationship between the cell type and the anatomic structure of the brain, I first group the cell clusters into major cell types. So based on the expression of well-established cell group markers. In total, 8 cell populations were identified, including glutamatergic neuron with different glutamatergic transporters, GABAergic neuron, astrocyte, blood vessel cell, microglia, oligodendrocyte, and oligodendrocyte progenitor cells. So in this figure, I plot the spatial distribution of major cell types on the left side and those of cell clusters on the right. I hope you can appreciate that major cell population illustrates the major structure of the brain. such as SLC17A7 glutamatergic neuron, are mainly distributed in the cortex and hippocampus, and the SLC17A6 positive glutamatergic neurons are mainly in the thalamus, with GABAergic neurons are enriched in the striatum area. On the other hand, the cell cluster on the right side with different gene expression represent fine anatomic structures. For example, you can see different cortical layers and different subregions of hippocampus. So to quantify the spatial relationship of different cell clusters, I utilize the spatial neighborhood enrichment analysis, and the results are shown as this heat map. On this heat map, there are two big blocks, A and B. So when plotting the cell clusters in each block on the tissue, you can see they are enriched in different parts of the brain. So this suggests a relationship between the molecular and spatial features of cell clusters, because the cell clusters is solely defined by the gene expression, but they also show very distinct spatial pattern. So if we take a close look at all different glutamatergic neuron clusters, we can find that different cell clusters clearly distinguish different cortical layers and hippocampal subregions. But in thalamus and hypothalamus and more posterior regions such as middle brain, no distinguishable cell clusters were identified for subregions in this first round of clustering. However, when I extract those clusters corresponding to those big brain structures and perform another round of clustering analysis, we can resolve more detailed cell types. For example, the original cluster 13, which corresponding to the whole thalamus, now is divided into multiple cell types. And you can see here are some examples showing different subclusters of the previous cluster actually occupy the different subregions of the thalamus. As mentioned earlier, in addition to genes for cell typing, the 1000-plex brain gene panel also includes many functional relevant genes, especially those encoding ligand and receptors. By calculating the expression level of different ligand and receptor gene across cell clusters, we can infer the potential cell-cell interaction among different cell types. Notably, taking the spatial distribution of cell cluster into consideration can help us better assess the interaction, which are solely inferred from gene expression. For example, here are cells express EDN1 genes, which encode the ligand endothelin-1, which can send signals to its nearby cells, which express two receptors of EDN1, which are EDNRA and EDNRB. As another example, here I show a VIP positive interneuron can send signal to its adjacent cells, which are express two different VIP receptors, which are VIPR1 and VIPR2. Overall, with the 1000-plex MERSCOPE gene panel, we are able to achieve high accuracy and detection efficiency within mouse brain samples. The data not only detailed cell typing, but can also provide spatial information of each cell type and can help us to gain functional insight into different cell types as functional relevant genes are included in this assay. Next, I'm going to present the data generated from human brain with 1000-plex gene panel. In this work, I perform multi-omic profiling of cortical slices from both normal people and AD patients, aiming to identify cellular and molecular changes associated with AD pathogenesis. So to design the 1000-plex gene panel used to study AD in human brain, I select two groups of genes. One group of genes for cell typing, and the other group of genes are implicated in AD pathology based on previous literature. For cell typing, I select cell type markers based on large-scale single-cell RNA sequencing and morphic data generated from human brain, which contribute to around 500 genes. So to select the AD-associated genes, I included genes involving many different signaling pathways which have been linked to AD, such as A-beta processing and phosphorylase tau. You also include pathways like neuroinflammation, microglia activation. This group of genes also contribute about half of the gene panel. With these two group of genes, we should be able to assess AD-associated gene expression changes in a cell type-specific and spatially dependent manner, which will demonstrate the stress of large gene panel on studying complex biological processes such as neurodegeneration. So in total, 4 datasets have been generated with brain slides from 2 human cortical regions, 1 control and 1 AD, with each have a replicates. So the most of data from replicates again show very good correlation, indicating high reproducibility. We also compare the normal brain data with the bulk RNA sequencing data from the normal human brain, which all again indicate a high accuracy. On average, in the whole dataset, we get about 50,000 cells, with each cell have around 600 transcripts detected. So unbiased clustering analysis of the cells from all the samples identify 22 cell clusters with distinct gene expression profile. The spatial distribution of these cells clearly illustrated an anatomic structure of human cortex, such as different cortical layers, gray matter, and white matter. So to gain a deeper understanding of the gene expression and spatial features of cell types in human cortex, I performed another round of clustering analysis with glutamatergic and GABAergic neurons. Here I first show the result from glutamatergic neurons. So specifically, I can identify 15 different excitatory neuron subclusters, each with distinct gene expression pattern. Here I show 3 examples which showing the core expression of the cell type specific markers such as POL3F1, SYT6, and PLCH1 with the pan-glutamatergic neuron marker SLC17A7. As expected, so different glutamatergic subclusters are organized into different cortical layers. Consistently, the neighborhood enrichment analysis showed that different subclusters are divided into different subgroups, with the cells in each subgroup are close located to each other, but relatively far away from the cell types out of that group. So conventionally, people using some established marker to distinguish different cortical layers, such as the CUX2 are enriched in the superficial layer, and this FISF2 are enriched in the deep cortical layer. Here we see a high consistency between those previous knowledge with our Murphy data. For example, both class 3 and class 5 are highly expressed this COX2 gene, and they are located in the superficial layer. But interestingly, we can see between these two, they still show some different spatial distribution in this human cortex. On the other hand, the class 1 and the class 7 are highly expressed FISF2 gene. Again, they also show some difference between their gene expression and the spatial distribution in the Murphy data. That suggests that the Murphy data can further distinguish like those subtle difference and identify fine cell types in human brain. So similarly, I performed the subclustering analysis with the GABAergic neuron, which revealed 11 different subtypes. So plotting the conventional interneuron subgroup markers such as PVLB, SST, VIP, and LAMP5, you can see the cluster results from Murphy's are well aligned with these different groups, but we just get more fine cell types. These are also co-plot the pan-GABAergic neuron marker GAD1 with different markers of subclusters. We are looking at the spatial distribution of these different GABAergic neurons. Most of them are just evenly dispersed across the whole cortical region, but there's one exception, that is cluster 8. You can see this cluster is heavily enriched in the very, very superficial layer. That's actually layer 1. So this analysis of both GABAergic and glutamatergic cells demonstrate the capability of MERSCOPE data in resolving gene expression and spatial organization in human brain. So based on the result of cell typing, we could further assess the gene expression change in a cell type specific manner. Before that, I would like to mention a unique feature of the MERSCOPE platform, which is its ability to perform protein co-staining with transcriptomic profiling. This is especially useful when some features could not be detected in the form of A. In this case, I include two antibodies to stain. The two features are highly relevant to AD, which are A beta plaque and hyperphosphorylated tau. So in the brain slides from AD patient, we can see robust A beta plaque and the p-tau signaling be detected by antibody staining. So in this enlarged picture, you can see the expression of glutamagic neural marker SLC17A7 and the microglial marker MOG. In addition, we can also see the A beta plaque and p-tau staining. So including these protein pathological markers can enable us to perform some additional analysis in addition to RNA, which I will present later. So the first comparison I did between the normal and AD brain samples are the gene expression change in different cell types. Here, I first group different cell clusters into 7 major different cell types to get enough cells, and then perform a differential analysis within each cell population between the normal and the AD samples. You can see in different cell population, we can detect a different number of differentially expressed genes, with microglia as the most affected one. On the right side, I did a gene ontology analysis, basically to identify the signaling pathways that are enriched in these differentially expressed genes. This analysis is done with the differentially expressed genes from microglia. You can see some I'm not sure you can read it. So, but pathway analysis from this differential expressions reveals some interesting hits, such as like modulation of chemical synapse transmission, regulation of neuron death, and the cellular response to cytokine stimuli, and such as microglia activation. So this has been heavily implicated in recent AD studies. So very interestingly, here we can see the glutamatergic neuron actually show relatively less differentially expressed genes. I think that may be because diversity of the cell cluster within this big group will impel the gene expression, as different subpopulations may exhibit different gene expression change. I will get back to this later. So with both gene expression and spatial information, I was able to analyze the cellular and molecular change associated with specific features. Here, I'm using the A beta plaque as an example. First, using the image of A beta plaque, I'm able to extract those A beta plaque positive regions and then get the coordinate of this region. Then we can calculate the distance of each cell to this A beta plaque region and generate this plot. So interestingly, when we calculate the cell type composition within different distance range to A beta plaque, we can see that neurons are relatively enriched in regions close to the plaque, while oligodendrocytes are more enriched in regions distant from the plaque. If we perform the same analysis with different glutamatergic cell clusters, we can clearly see that cluster 1 is enriched in A beta plaque region, while cluster 13 is depleted from the region. So based on the spatial distribution of cell cluster, we can further analyze the gene expression change associated with spatial distribution. For example, here I show the differentially expressed genes within the microglia cells, which are closely or far away from the A beta plaque. As another example, I calculated differential expressions across different cell clusters and see if there is a relationship between the number of differentially expressed genes and their spatial distance to A beta plaque. As you may remember, the cluster 1, which is enriched in the A beta positive region, actually show a high number of gene expression change compared to the cluster 13, which is enriched in A beta plaque depleted region. So very interestingly, in this analysis, we found the cluster 21 shows the highest number of differential expressed genes across all cell clusters, actually, which is not associated with A beta plaque. However, The spatial distribution of this cluster actually closely resembles that what we see of the phosphorylated tau staining, suggests the phosphorylation of tau may be a driving force of the gene expression change of this cluster. So altogether, the analysis indicated that different cell types, even in the same cell type, may differentially affected in AD brain, emphasize the importance of acquiring both spatial information and molecular information in study brain disease. So to summarize my presentation, today I show some data generated from the new 1000-plex MERSCOPE gene platform. This big gene panel enables precise spatial profiling of 960 customized MI species within intact tissue context. So I also present two showcases. One is applying the 1000-plex gene panel to mouse brain will not only achieve detailed cell typing, but also resolve anatomic and functional characteristics of different cell types. Also, when we apply the 1000-plex gene panel to human brain, we are able to profile the cell type and identify gene expression change associated with Alzheimer's disease. Finally, simultaneous assessment of mRNA and protein with the spatial organization of cell types within the human cortex, which can uncover the cellular and molecular change linked to Alzheimer's disease pathogenesis. I will end here and very happy to take questions. Thank you for your attention.
Well, thank you, Peter and Renchao, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. We've already got some great questions coming in here. This first question asks, is there a specific reason why you chose 960 instead of 1000 genes?
Yeah, Kaylee, so I can take that question. So 960 was selected in order to optimize the time that's required for running a 1000-plex experiment. Additionally, there are blanks that are included in the experiment as well. So those were the reasons why 960 was chosen over 1000.
Great, thank you. All right, Next question here. This is a two-part question. This asks, does cell segmentation account for diverse cell shapes, for example, axons of neurons that may be distal from the cell soma? And how does segmentation ensure the transcripts far from the cell soma get segmented with its original cell and not necessarily the nearest cell soma?
I'm going to take this one. So for now, we're still most assigned the transcript adjacent to the soma, to the segmented cell, because as you know, in the brain, the cell shape is very complex, especially including those axons and dendrites. And the cells sometimes, in most cases, are also packed very dense. So it's really challenging to segment those transcripts distant from the cells. And even with tracing, you can hardly identify those the axon or dendrite that's corresponding to each cell. There may be some specific labeling method. In that case, maybe we can trace some cell segmentation algorithm to do that. I think recently we kind of implement cell post2 in our segmentation tool. That means if the labeling is very sparse, like you include some fluorescent labeling, maybe you can train the algorithm to assign some distal transcript to the cell. But in most cases, we still rely on those transcripts adjacent to the soma. But in most cases, if we look at the transcript density, most of the transcripts are actually located adjacent to the or in the cell soma. So there's not much information loss in a normal brain data.
Great, thank you, Renchao. All right, next question here. Could you comment on the difference in acquisition time, for example, for the 1000 versus 300 gene panel, and the difference in cost?
Yeah, so I can take that one. So between our 300 and 1,000, it's a difference of just a few rounds of imaging, a few rounds of hybridization and imaging there. So it's going to vary, and it's going to vary based on the size of the tissue, but you could probably approximate about 8 to 11 hours difference between a 300 and 1,000 plex run. As far as price, the price for our 1,000 plex is in line with our current product offering. So from that 300 to 500 and 140 up to 1,000.
Great, thank you, Peter. All right, next question here. What is the maximum tissue region you can analyze per MERSCOPE reaction slide?
Yeah, so I can take that one as well. So it's
I can take that one.
Go ahead, Renchao.
Go ahead, Peter. Okay, so now the limitation is like 1 square centimeter. So as long as you can like put multiple tissues in on the same MERSCOPE slide, finally you can image it up to 1 square centimeter region. That includes multiple subregions if you can have multiple tissues.
Great, thank you. All right. Now, what is the size limit of tissue that can fit on the slide?
I can take this one. The slide, the MERSCOPE slide actually is much larger than the 1 square centimeter. So for example, if you have 2 to 3 square centimeter tissue, you can put on the MERSCOPE slide. But as mentioned, the final imageable area is kind of limited. For example, if you have a cancer tissue or relatively big, like some other like big brain samples, and you can put that in one MERSCOPE slide, but during the image, you can select multiple regions from that the whole tissue that up to 1 square centimeter.
Great, thank you, Renchao. All right, next question here. Are those AB plaques identified by protein or protein stain?
Yes, that's another unique features of MERSCOPE platform. Basically, we can combine protein staining with MERFISH experiment. So the A beta here is identified by a specific antibody.
Great. Now, do you recommend the use of spatial information when clustering?
That's a very interesting question. I mean, so far the analysis I did is kind of first just using the molecular information to do that, and then to assess the spatial relationship or the correlation between the spatial distribution and the molecular features. But I know there are some algorithms have been developed can using both. In that case, because in my experience, I always find these two kind of correlated. So another way to think about this is because after you did clustering, if you think there are some spatial features varied between this cluster, you can even go back, for example, to further divide this whole cluster according to the spatial information, and then further identify the molecular difference in these subgroups. That's really doable. So I think for now, the spatial genomics is also, the analysis is also like actively been developing. So probably like more exciting algorithm will be generated to kind of dig into the inside of this comprehensive data.
Great, thank you. All right, next question here. Could you please repeat how you stain for amyloid plaques in mouse and human brains?
Oh, so we have a kind of developer protocol to combine the protein staining with MERFISH. Basically, we just add in the antibody staining step within the workflow of sample prep. So the antibody, it's we can like use different we support different antibodies from different species, like rabbit, mouse. The antibody, the primary antibody is just based on what you have been interested, and then we have like a solution to detect those primary staining. So that's already implemented in our workflow.
Great, thank you. All right, next question here. How does transcript detection specificity compare in the 1000 plex panel versus the 500 gene panel? And do you lose some specificity when going up to 1000 genes?
So we do some comparison between the data generated between the 500 gene panel and the 1000 gene panel. If we just, for example, overlap, do two experiments with the different gene panel and then compare the counts on the same tissue, we don't see like significant difference between the two data sets. Basically, the detection efficiency almost maintained. Also, the specificity, because we can compare kind of the correlation between the overlap between the two panels, we almost get a linear relationship. So we think the specificity can also maintain.
Great, thank you, Renchao. And I want to thank our audience for all these great questions coming in here. We do have some more time, so we will continue with a few more here. Our next question asks, how many antibodies are available and validated now for co-imaging with 1000 plex RNA MERSCOPE imaging?
Here we haven't specific validated antibodies with the 1000 plex gene panel, because in the workflow, although we can do this together, they kind of separate from each other. We just using like additional image rounds for the antibody staining. So because we have validated many antibodies with, like in our previous development, with relatively less gene panel, so we don't think or we don't see any issues like applying this already validated antibody to the new 1000 plex gene panel, because they kind of the workflow is still the same.
Great, thank you. All right, another question here. How did you do the cell annotations? It looks like the cell annotations are very accurate.
So there are different ways to do cell type annotation. One way is basically manually do the annotation. I still consider that's the most accurate. The strength of MERFISH or MERSCOPE is that you get both molecular information and the spatial information. Combine these two is relative. To me, it's quite simple to annotate those cell types. Sometimes even the cell type marker, like the molecular marker, is not that obvious. But when you see the cell type distribution, usually you will get a very good idea what this cell type could be, because sometimes it's only with the molecular marker. Some cell types may share the markers, but if you combine the molecular information and the spatial information, I think it's quite easy to annotate the cell type. And if you want to use some automatic annotation, that's also doable. I think there are some atlas or like reference data have been generated, and some algorithms can automatically annotate those cell types based on molecular information. But I think it's always helpful to check the annotation by plotting the cell spatial distribution, and then can do some manually adjustment if you think that's not correct. Yeah, that's my answer.
Wonderful, thank you. All right, next question asks, can you design a way to identify or mark the region we could apply on the MERSCOPE slide?
Yeah, so I can take that question. So we're actually getting ready to launch a solution around that. So it'll be a sample placement guide that you will attach to the bottom of the MERSCOPE slides. And this will allow you to visualize where that imageable area is on the MERSCOPE slide and just more accurately place those tissue samples when you go to mount them.
Great, thanks, Peter. All right. Next question. If you include cell bounds in all aux channels, what are you seeing the maximum runtime being with the 1000 plex?
Yeah, so I can take that one as well. So maximum runtime, if we max out all of those settings there, it's approaching 65 hours, which again, that's maximum tissue area, cell boundary stain in all of the auxiliary channels, which is, you're getting a lot of information there. So yeah, it approaches 65 hours, but nothing absolutely crazy there.
Great. Now, how many proteins or antibodies could you stain?
So far we support up to 6 different antibodies combined, but they need to come from different species.
Great, thank you. All right. Do you observe reduced expression broadly in cells nearby plaques?
That's actually a very interesting question, because I think so far I didn't do that specific analysis, but definitely I can look into that. Currently, I'm doing the differential expression analysis. I think that's actually a interesting point, because the cells may be not that healthy in that area. So even in the same cell type, the transcript density or compile the same genes, we can maybe see some difference. Thanks.
Kaylee, I think you're muted. We can't hear you.
Sorry about that. Thank you. I was thanking you again, Peter and Renchao, for your time today, as this will conclude our session. So before we go, do you have any final comments for our audience?
Yeah, so just want to mention again that both the MERSCOPE 1000 gene panel and MERSCOPE 1000 gene imaging kit are available for order now. Yeah, so we're super excited for that, and we think this is going to be a great solution for the MERSCOPE platform.
Great. Well, thank you again, Peter and Renchao, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Vizgen, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.